First off, I would like to thank the organizers for putting this meeting together. It's a new format, but I think it's very important to give us the chance to share our results with the scientific community. And let me announce the first presenter. So the first presenter is Kia Markussen, okay, from University of Kentucky. And the title of her presentation is Lafora disease, differential metabolic disturbance in neurons and astrocytes. Kia, you have 12 minutes.
Thank you for the introduction. It's my pleasure to present today at this online webinar. And my name is Kia, as you just said. I'm from the University of Kentucky, and the topic is Lafora epilepsy. So LD, or Lafora disease, is a fatal autosomal recessive disorder characterized by epileptic seizures and accumulation of insoluble glycogen-like inclusions that we call PGBs. And it's classified as a glycogen storage disease. The symptoms typically manifest themselves in the second decade of otherwise normally functional kids, and they rapidly progress with this first epileptic seizure, and then rapidly they experience neurodegeneration and ultimately die typically within 10 years of the first symptoms. The disease is caused by mutations in the genes coding for either Laforan, which is a glycogen phosphatase, or Malin, which is the E3 ubiquitin ligase. And these mutations can lead to this aberrant glycogen metabolism and LD. And we do, this is a loss of function, sorry. And we have mouse models for both of these genes, the Laforan and Malin knockout mice, and today I will focus on the Laforan deficient mice. So the community has done a great job of characterizing these hallmarks of the disease. And we know, of course, here the PGBs, which have been established to drive the disease. But other than that, we have another range of hallmarks, including impaired mitochondria, glutamate metabolic transport, and neuroinflammation. One thing that's important to remember is that early on, it was established that PGB accumulate in neurons, and the glial cells will largely ignore it for the first period. However, we do know today that the glial cells as well do accumulate glycogen, the PGBs, and they are different from the one accumulating in the neurons. So therefore, we asked ourselves, how is the metabolism impacted in specific brain regions, and how are the cell types impacted? So one of the first things we looked at was where these PGBs accumulate. And here we observed that early on in the disease state, we have an accumulation in the hippocampus. So today I want to focus on the cortex and the hippocampus. And we know that the hippocampus is associated with epilepsy and therefore likely important in LD. We know that there are two major cell types in the brain, the neurons and the glial cells. And today I want to focus here on the astrocytes, which are often described as supportive cells. And they take part in clearing glutamate from the synaptic cleft and excess potassium. What all of these cells have in common is that they need glucose to function. And as we know, it can be degraded to pyruvate through glycolysis and into the TCA cycle as acetyl coenzyme A, and then generate this proton gradient that can be converted to ATP. However, this is not the only energy substrate in the cell. And the important amino acids, glutamine and glutamate, can be utilized as well as energy substrates. And the glutamate released from the synaptic cleft can be taken up by astrocytes, then converted to glutamine to release back to the neurons, or they can enter the TCA cycle as alpha-ketoglutarate and used for energy production. And it's important to know that even small disturbances in energy homeostasis can be fatal. So we want to look at how we can quantify metabolites from different pathways. And one way we can do that is by gas chromatography mass spectrometry, and we do that in our lab. Here we pulverize the tissue, and extract the polar metabolites, and then derivatize with an agent called MSTFA. Then we obtain the mass spectras and do the data generation by matching it to a library of standards. And we know one thing is important is that a mass spectrometer can measure very small changes in mass, such as 1 proton or neutron. So we take advantage of carbon isotopes, stable carbon isotopes. And we know 99% of all carbon is found as carbon-12. But we can also generate carbon-13, as we have here with the glucose or glutamine or glutamate. So we substitute all the carbons for carbon-13 and then incubate the cells with these substrate. And then from here, you will have this labeling patterns arising where initially two carbon atoms are labeled with 13C, and we can measure the decomposition all around in the intermediates in the TCA cycle. And similar, if you incubate with glutamine and glutamate, you'll get this labeling patterns of M+4, means all of these are have 13C labeled carbon. So the only thing important to remember here is that different substrates will generate different labeling patterns that we can measure by GCMS. So first, we want to look at glucose metabolism in these brain slices. And we can then cervical dislocate these animals, dissect the cortex and the hippocampus, and use a Magellan tissue slicer to give us these brain slices, and then incubate them and with 13C labeled glucose and examine the labeling patterns. And if we see here, you have the cortical brain slice on top and the hippocampal brain slice on the bottom. And you see here a decrease in glucose metabolism as a decreased lactate and several of the TCA cycle intermediates. And especially here, the hippocampal brain slices appear to be vulnerable. So overall, we found a decreased glucose metabolism in Lafora deficient animal in both cortex and hippocampus, which is expected. Since this is a glycogen storage disease, we expect to find disturbances of glucose metabolism. Then we moved on to glutamate metabolism, which we found to be largely unaltered in both brain regions. We see here no differences in the labeling patterns whatsoever in either region. Therefore, we think that these animals have a normal glutamate metabolism. Then we looked at the glutamine metabolism. And here we see glutamine is primarily metabolized in the neuronal compartment. And we observed this drastically increase in glutamine metabolism in both brain regions. And therefore, we know, so what did they say? Glutamate is primarily metabolized in astrocytes, and glutamine is primarily metabolized in the neurons. So from these data, we observed these differences in cell-specific metabolism, and we wanted then to see if we can trace this back to these cell types. So therefore, I generated neurons, primary cultures of neurons and astrocytes, and similarly incubated with glucose and glutamine and glutamate, and analyzed the incorporation. So we first look here at the primary culture of neurons. You see a slightly decrease in neuronal glucose metabolism, which is replicating the things we saw in the slice data. However, when we looked at the astrocytic glucose metabolism, we see an increase in glucose metabolism, which is surprising since as you remember, the glycogen is primarily stored in astrocytes. So overall here, we see a decreased neuronal, replicating the slice data, but an increased astrocytic glucose metabolism. So we moved on to glutamate and glutamine metabolism. And here we incubated the primary neurons with glutamine, and we saw a slightly increased glutamine metabolism. Therefore, meaning that this is again replicating the slice data, and likely the increased glutamine metabolism coming from the neurons. Strikingly then, when we looked at the astrocytic glutamate metabolism, we observed a drastically decreased glutamate metabolism in the astrocytes, which is not the same picture as we saw with the slice experiments. So overall, a drastic decrease in glutamate metabolism in Laforan deficient astrocyte, but increased glutamine metabolism in the neurons. So with that, lead us to the conclusion that we here first observed a hypoglucose metabolism, which is likely coming from the neuronal compartment, but an increased glucose metabolism in the astrocytes. And this is in line with what we know from other neurodegenerative disorders. We see a hypoglucose metabolism. And when we looked at the glutamate metabolism, we saw drastically decreased glutamate metabolism in the astrocytes, but no major changes in glutamate metabolism in the brain slices. So perhaps in this early time in the disease, the cells are compensating, perhaps the neurons are taking up more glutamate, or the microglia could take part in to clearing this glutamate from the synapse. And we know that the GLT1 transporters have shown to be impaired in astrocytes. And finally, we observed an increased glutamate metabolism. And this is perhaps a possible compensatory mechanism by the neurons to compensate for decreased astrocytic glutamate and glucose, disturbed glucose metabolism. We saw this increased glutamine metabolism in both brain slices and neurons. And there's actually a growing body of evidence showing that epilepsy and dysregulated glutamine metabolism associated, but it's not straightforward. And it's not something we know a lot about in LD yet. But it would be interesting to see the connection between glutamine dysregulation and LD and glycogen. So with that, I'll say we showed differential metabolic disturbances in both LD brain regions and neurons and astrocytes. And with that, I want to thank my current lab, the Differential lab. We have close collaboration with the Sun lab, and my former lab, the neurometabolism lab at the University of Copenhagen, where I was when we started up this collaboration, and of course our funding. And with that, I will be happy to take questions.
Thank you, Kia, for your presentation. It's a lot of work, and it was beautiful work done. So we have a bunch of questions here. Let's see if we can actually answer some of them. So the first question is is a glutamate metabolism unaltered in the neuron cells, or is it altered?
Sorry, can I see the... Oh, I'm sorry. I just want to see the questions.
Yeah, so what happened with the glutamate metabolism in neuron cells?
In neuronal cells? So I did not see any changes in the neuronal metabolism for glutamate at all. There's have been no evidence so far.
Okay, so another question was, did you check for GLUT3 and 4 in the brain samples?
Mm-hmm. That is an excellent question. There is almost no, we haven't done it yet. I would love to do it. There's only one question, one paper out there that slightly suggests that there might be something wrong with the glucose transporters, but it's not something we've chased yet.
Okay, I have a quick question for you. So you said there are also mitochondrial defects that you observe in the patients. Can you actually build up a little bit on that? What kind of mitochondrial defects those patients have?
So one thing that's been done is look at the mitochondria seems to be fragmented. But from the other experiments I've done, we looked at mitochondrial stress test and glycolysis stress test. We don't really see big changes. So it's hard to say exactly what's going on, but they appear to be fragmented. Yeah.
Okay, one last question. What are the distributions between neuron versus astrocyte in cortex or hippocampus?
That's a really good question. So, and a little hard to answer. So previously, it's been said that there were way more, like a tenfold more astrocytes in the brain than neurons, but more recent studies actually showed it's more a one-to-one ratio. So.
Okay, we do have more questions, but I'm afraid we don't have time to go over the other questions. I want to thank you very much for your presentation. Great work. And with that, so we can go for our next speaker. So our next speaker is Stephen Williams, okay, from Meharry Medical College. And the title of his presentation is Hypoxia-Induced Annexin A6 Expression Underlie the Metabolic Vulnerability and Response of TNBC cells to EGFR tyrosine kinase inhibitors. Stephen, so you have 12 minutes.
Thank you, everybody. And as Dr. Tortellotti has announced, my name is Stephen Williams, and I am a PhD candidate at Meharry Medical College. And so today's talk is going to revolve around the health disparity amongst the triple-negative breast cancer subtype. And so we know that breast cancer is the most common cancer type in the United States amongst women, and it's the second leading causes of death, second to that of lung cancer. And so what the field has done is that you can molecularly subtype breast cancer into the normal, luminal A, luminal B, or HER2. And this classification is based off of the hormone receptors, estrogen and progesterone, or the human epidermal growth factor-like receptor, or HER2. And so in triple negative, which the name deems, these particular subtype of breast cancers are negative for your hormone receptors, but they are also negative for this growth like receptor HER2. And it accounts for 15 to 20% of all breast cancer subtypes. But even within triple negative breast cancer, you can then classify it into 4 different distincts: immune basal-like 1, immune suppressed basal-like 2, your mesenchymal-like, or your LAR, your luminal androgen receptor expressing group. And so my lab and the Sakwe lab at Meharry, we focus on this basal-like TNBCs. And some of the characteristics is that they overexpress or hyperactivate the epidermal growth factor receptor, also known as the EGFR, which is a common oncogene in a lot of cancer subtypes. They have high mutant p53, which is a well-known tumor suppressor. And they are highly aggressive, prone to metastasis, and therefore they have this very poor prognosis. And what our lab actually has shown is that they develop resistance to this EGFR TKIs. And what makes it a health disparity is that the Kerry group in 2006 were the first to show that this particular subtype of triple negative breast cancer is actually more prevalently found and diagnosed or prognosed in the African American population compared to that of other ethnic groups. And even amongst that, it's in the younger African American as depicted in the schematic. And so the field of breast cancer is very complex because these tumors are very heterogeneous. And so within TNBCs alone, you have this subpopulation of cells that are rapidly growing, or these basal-like cells, or you can have these cells that are highly invasive, or those metastatic cells, and mesenchymal-like, but both lead to this type of resistance to this therapy, which make them hard to treat tumors. And so what our lab is trying to do is trying to understand what are some mechanisms of this resistance. And so we focus on the EGFR, so also known as the epidermal growth factor receptor. And so this particular receptor is a unique class of receptors, a part of the ERB family of tyrosine kinase inhibitors. And so upon dimerization, it could be either homo or hetero. You can have the receptor at the extracellular domain have these growth factor ligands which can bind to it, and in the intracellular part, you have this tyrosine kinase domain where they will autophosphorylate the other receptors, leading to the stability of this EGFR receptor and the downstream signaling pathways, PI3, AKT, MAP kinase pathway, and your JAK-STAT pathway, which ultimately lead to tumor cell growth, migration, and invasion. Well, in the field, this is a very elucidated pathway because, like I said, it's overexpressed in a lot of different tumors, making it therefore a oncogene. And so in the field of breast cancer and in lung cancer and colon cancer, you can target it in the extracellular domain using these monoclonal antibodies, such as cetuximab in non-small cell lung cancer, or you can inhibit it at the TKI or the tyrosine kinase domain using lapatinib or lotinib, gefetinib, which have been FDA approved for the use of non-small cell lung carcinoma, and as well as in some colon cancers. However, for the use of breast cancer, we see that these patients within a short amount of time develop this resistance to this particular therapy, and we're trying to elucidate mechanisms why. And so one of the interesting things my lab has been able to identify is that EGFR is stabilized or co-localizes to this plasma membrane region in that of enriched in cholesterol, also known as these lipid raft regions. And so one of the things we believe that is actually a mechanism of this resistance is the belief of hypoxia in the core of tumors. And so hypoxia by definition is the deprivation or the lack of oxygen in the vasculature. And so what research has shown is that this hypoxic tumors or hypoxic regions of tumors actually sensitize the breast cancer cells that overexpress EGFR to this TKI. And as this schematic is showing, is as you go deeper into the core of a tumor, it becomes more hypoxic, leading to also necrotic tissue or death. And so one of the phenomenons of this particular hallmark of the tumor microenvironment is the transcription factors, hypoxia-inducible factors, which actually drive this ability of these hypoxic viable cells to actually more so undergo metabolic vulnerabilities and or reprogram. So therefore leading to the alteration or the manipulation of different genes involved in cancer progression. And so what our lab is elucidating is this particular tumor suppressor known as annexin-A6. And so annexin-A6 is a part of a annexin family of genes, but it's interestingly that annexin-A6 has 8 annexin repeats compared to that of the 4 of the other annexin families. And it also has more calcium binding sites. And some of the functions that have been investigated, not by just the Stockwell lab, but in the field, is that annexin-A6 has a plethora of functions, including calcium homeostasis, endo and exocytosis, cholesterol homeostasis, such as the lipid raft domains. And our lab has shown that reduced expression of annexin-A6 actually promotes the rapid internalization and therefore the degradation of this EGFR, and thereby it sensitizes it to these TKIs. And so going back to the whole story of annexin-A6 and breast cancer, it was actually first elucidated from our lab in 2011, where we showed that annexin-A6 expression actually correlates to this invasive or this breast cancer phenotype. And so that cells that express high levels of annexin-A6 actually take on this invasive property, but when you downregulate annexin-A6, you inhibit that invasive property, but you see the rapid growth or the decreased migratory ability of these cells. And then followed up in 2013, our lab actually also shown that when you take the cells that do express annexin-A6 and you downregulate them, you actually change their cancer phenotype. And it actually shows that when you downregulate annexin-A6, these cells are thereby show a rapid growth and their migratory or invasive property is depleted. But interestingly, we see that these downregulation of annexin-A6 is sensitive to these EGFR approved TKIs, and bringing your attention to lapatinib and PD-153, which were the most sensitive compounds used in this particular study. And then we also showed in that paper that in you're looking at a Kaplan-Meier plot of over 3,000 breast cancer patients, that we show that the annexin-A6 expression is not significant in the correlation to the relapse-free survival, distant metastasis-free survival, or the overall survival of these patients. However, in B, in particularly the basal-like subset of TNBC breast cancer patients, you actually see that low levels of annexin-A6 is significantly correlated to the higher relapse-free survival. And on the contrary, that high levels of annexin-A6, is actually correlated to higher distant metastasis-free survival and an overall survival in these patients compared to that of annexin low cells. And so then we followed it up in vivo, where we took these same cells where we downregulated annexin A6, and we saw that in cells that express this low level or down inhibition of annexin A6 upregulation, you saw the rapid growth of these xenograft tumors in these null mice. And you see in BT-A6A, where we actually knocked out annexin A6 in the prior 2011 paper, by day 18, we had to sacrifice these mice because the tumors were so large. But then in that same study, we showed that if you take these now HCC1806, a basal-like cell that expresses endogenously low levels of annexin A6, and you overexpress annexin A6, you actually see the reverse, that in cells that in mice transfected with these empty vector cells compared to that were overexpressing annexin A6, you see a decrease in tumor growth. But what we also saw in that paper when we did this mass spec analysis is that the cells that when you downregulate annexin A6, there were a plethora or a large amount of genes that were actually upregulated. And in those genes were some key genes, such as your cadherin-2, your JAGAT1, your RAS-GRF2, and your fatty acid binding protein, all genes that are involved in the differentiation, metabolism, and growth of these TMBC cells. And then we did a KEGG pathway analysis, and we actually see that when you downregulate annexin A6, all these particular pathways are upregulated, and bringing your attention to the ones indicated in the box. And my dissertation work is focusing on the energy metabolism. How is it that these cells are actually being much more rapidly growing and leading to this rapid proliferation? And so we gathered the hypothesis that in these annexin A6 high cells that have this invasive or metastatic potential and the downregulation of these different genes described in the prior slide, but when you downregulate annexin A6, you see that these cells are able to rapidly grow in these mice, as well as in these 3D matrix gels. They are identified by aggressive tumors, which leads to the metabolic switch or the metabolic differentiation of these key genes. We are hypothesizing, and we strongly believe that hypoxia, tumor hypoxia, could be actually modulating the annexin A6 expression in these cells. So the first thing I wanted to do was to actually take BT549 cells, a mesenchymal TMBC cell-like, and MDA468, a basal TMBC cell-like, and induce them to 1% oxygen in a backstroke hypoxia chamber over the course of 4 days. So we covered acute, which is in literature 24 hours or less, and chronic, which is that greater than 24 hours. And as you can see in the BT549 cell lines, annexin A6 expression is much more stable, readily stable, but you do see the upregulation by day 4, but you see a more pronounced stability of this hypoxia-inducible factor 1A in BT549 cells compared to that of the MDA468 cells. After day 1, you see HIF1 stability is actually lost. And when you check the mRNA at day 4, so this chronic exposure, you actually see that annexin A6 is up compared to normoxic conditions in both cell lines. And we wanted to make assessment of some HIF1 alpha downstream target genes such as GLUT1 and PGK1, which are both glycolytic enzymes involved in glucose metabolism. And so we see that annexin A6 is upregulated. We wanted to see, well, what happens if you inhibit annexin A6 upregulation in these cells? How do they respond to this hypoxic induction? And so what we did was we took BT549 cells that were transfected with non-silencing control and that of your SH5 short hairpin RNA targeting annexin A6. And what you see is that even under hypoxic conditions, that when you inhibit annexin A6 expression, you actually lose HIF1 and HIF2 alpha stability. However, in this MDA468 cells that by day 4, they no longer express the protein expression of HIF1 stability, we monitored HIF2 alpha stability, and it's been shown in literature in this particular cell line back in 2000 that HIF2 alpha has this endogenous expression in this particular cell line, even in the presence of normoxia. And we saw the same thing. Even in the presence of hypoxia, you don't see HIF2 alpha there, but when we actually transfect the cells with this empty vector and with a plasmid that overexpress annexin A6, you see that in the last panel, you were actually able to render, or this HIF2 alpha protein stability was actually recovered when you overexpress annexin A6. So this showed us that maybe annexin A6 and HIF1, HIF2 alpha actually has some type of correlation and are ultimately involving the
Listen, you have 1 minute.
Okay. And so then we wanted to look at some of the metabolic vulnerabilities that were found in these cells. And we see that under chronic hypoxic condition in the annexin A6 expressing cells, there's a much more pronounced reprogramming of these particular cell lines compared to the MDA cells that express endogenously low levels of annexin A6. And so we took it a step further and we looked at live cell measures, and we see that in both annexin A6 low cells, 468 and HCC70, they are actually using their mitochondria or their oxidative consumption, and they have much greater extracellular acidification, which could be from glycolysis and or mitochondrial respiration. And so then we wanted to go a step further and look at the response of these cells to lapatinib. And we see that in annexin A6 high cells, they are much more resistant, even in the presence of hypoxia. And we took it a step further in looking at lipid metabolism, and that when you downregulate annexin A6, there's a reduced accumulation of lipid droplets. But when you actually treat the cells with lapatinib, we see this rendered effect. And so overall, my particular project is looking at how does hypoxia manipulate or lead to the vulnerability of these particular TNVC cells, and how does annexin A6 play that involvement. And so I give acknowledgements to the Sockway Lab and to our collaborators, the Rothno Lab at Vanderbilt University, and I will entertain any questions at this time.
Thank you, Stephen, for your presentation. Beautiful work.
Thank you.
I have one question here. Have you guys looked at the promoter of annexin A6 and see what factors bind in that promoter to have an idea of annexin A6 regulation at promoter levels?
Yes, we have. That work is actually, we're planning to submit it within the next couple of weeks, and we actually do see that HIF1 and HIF2 alpha have a binding site within the annexin A6 promoter. Good question.
So another question, have you guys measured the ATP levels in the annexin A cells to see if it changes over time compared to the controls?
No, we have not done that yet, but that is lined up in the next set of experiments once we're able to reconvene at school. Good question. My committee asked that.
Okay, thank you, Stephen.
Okay, then it is my pleasure to introduce our final speaker, Giovanni Tortellotti from Tulane University, and I will give you the screen and time now.
Thank you, Kia. So good afternoon, everybody. Once again, I want to thank the organizers for putting this meeting together and allow us a chance to share our data in these difficult times. So before I start to present my data, I'd like to give you a quick background on kidney development. Kidney development, it's a very complex process that involves the interaction between different tissues and signaling molecules throughout the development of the organ. So, and these interactions can be actually better appreciated in small units that give rise to the mature nephrons over the development of the kidney. So now if we look at here on the right side of the screen, I have one of these units. So these units comprise three different tissues, three main tissues. In yellow, we see the ureteric bud, or UB for short, that gives rise to the collecting duct system in the kidney. In green, we see a very specific type of mesenchyme, the cap mesenchyme, because it caps the tip of the UB and contain the nephron progenitor cells, the cells that give rise to the nephron itself. And in dark blue is the embryonic stroma that gives rise to the cortical stroma and the medullary stroma. So there's a lot of interactions in between these tissues and secretion of growth factors and other molecules that modulate the development of the entire organs. And this is a well-accepted idea in the field. So recently, we've seen that all the molecules may play a role for cell fate decision in the kidney. In 2019, our lab published a paper showing that downregulation or inhibition of glycolysis use a pharmacological agent, YN1, that inhibits glycolysis at the level of PFK, drives cells of the developing kidney into the differentiation pathway. And this is better appreciated in the middle panel here, compare control with the glycolysis inhibitor figure, where we see a lot more of the red structures here. And the red structures mark the LHX1 positive cells, and LHX1 is a marker of differentiation in the kidney. So clearly, the glucose metabolism influence cell fate decisions in the kidney. So the question that emerged from that work was, what is the mechanism, how those glucose metabolism modulate cell fate decisions in the developing kidney? To answer these questions, we dissect those small units that I talked to you about early in the presentation at embryonic day E13.5, and we culture the NPCs, the nephroprogenitor cells, with different culture conditions. First control, and then YN1, okay, the glucose inhibitor, and KIR. Now, KIR is a pharmacological agent that promotes the activation of the canonical Wnt signaling. And the canonical Wnt signaling, it's one of the main signaling pathways that drives differentiation in the developing kidney. So it's a good positive control to compare with YN1. And we did this comparison by using proteomics and transcriptomes techniques. So at protein level, what we saw was at protein level, the topmost modulated pathway common to both treatments, KIR and YN1, are metabolic pathways. As seen here, the number one topmost was cholesterol biosynthesis. And I'm actually, I want to call your attention about cholesterol biosynthesis here, fatty acid metabolism that I've been talking about throughout my talk. And this is not different if you look at RNA levels. Also, metabolic pathways, they pop up as one of the top most modulated pathways regulated by both treatments, KIR and YN1, which are, again, two very different drugs. And again, at RNA level, we also see modulation of the fatty acid synthesis metabolism pathway and the cholesterol metabolism pathway, right? So, but what is the relationship between the cholesterol, fatty acids, and glycolysis? Well, in the developing kidney, those progenitor cells, they get super high in glycolysis, right? And increasing glycolysis leads to increasing the pyruvate masses. Now, pyruvate can be either converted into lactate or go into the mitochondria, and it's converted into acetyl-CoA. That was mentioned before. Now the acetyl-CoA feeds into the TCA Krebs cycle and gives rise to a number of metabolites and ATP the cell uses for a number of reactions. But also in the acetyl-CoA can be converted as first step of the cells of the TCA cycle into citrate, and the citrate is transported out of the mitochondria. And the citrate is now converted back to acetyl-CoA for a specific lyase. Okay, and this acetyl-CoA now feeds into fatty acid synthesis pathway or mevalonate cholesterol pathway, or the acetyl-CoA also serves as a substrate for lysine acetylation, histone modification, which are well-known mechanisms to control transcription in the developing kidney. So we first decided to take a look at these two main enzymes that are involved with the transport of citrate into the cytosol and the conversion of citrate into acetyl-CoA. And as we see here in both RNA and protein, in both treatments, YN1 and KIR, we see a downregulation. And we observe that with the transporter and the lyase. So it seems that the glycolysis inhibition, as well as activation of the canonical pathway, promotes a downregulation in the main enzymes that are involved with acetyl-CoA metabolism outside the mitochondria. So let's now take a look at the one of the branches I talked to you about early, which was the cholesterol metabolism. When you look at, this is proteomics, okay? When you look at many enzymes, several enzymes involved with cholesterol metabolism, pathway in the cells, we saw a downregulation with both treatments, YN1 or KIR. So both treatments, though very different, promote the same results, a downregulation in the cholesterol. And you can, and this can be seen here in the heat map and also in the bar graph on the side. So we ask ourselves, what's the importance of the cholesterol then for the kidney development? And we decide to take approach, ex vivo approach here. And we dissect and collect embryonic kidneys at day 12, E12.5, and we treat them for 24 hours in the presence or absence of pravastatin, which are drugs that inhibits cholesterol metabolism. And as you see here, and this can be better appreciated in the middle panel, so you see here, there's a lot less of the LHX1 positive structure in the control than in the pravastatin treated group. So that means that, again, a blockage in the cholesterol metabolism also drives cells into the differentiation pathway. That's exactly what we've seen before with the glycolysis inhibition. So, but as I said, so acetyl-CoA, it's a key molecule that links glycolysis with cholesterol biosynthesis, right? So we ask ourselves whether or not how acetyl-CoA, or in this case acetate, so we provide acetate to develop kidney, how does it affect the development of the embryonic kidney? And when we treat, again, E12.5 kidneys for 24 hours in the presence of 10 millimolar of acetate, what we see, it's an increase in the red stain here, the cap mesenchyme. We see it's a bigger cap mesenchyme with more cells in it. So overall, the kidney actually looks pretty good. So with acetate supplementation. So then the next question was, can we prevent the YN1 effects in these kidneys if we supplement it with acetate? So with acetate supplementation, prevent the YN1 effects? And the answer for this question was yes. As you see here in the first panel on the left side, so kidneys that are treated with YN1 in the presence of acetate, they have a much bigger cap compared to the top panel, a much bigger cap with a lot more cells. So it makes sense to us because the YN1 blocks glycolysis at the PFK level. But now if I provide acetate to the cell, I'm bypassing the glycolysis block and providing the cell with the substrate it needs to move forward with the development. We also saw that there's more nascent nephrons in this kidney, but again, the kidney overall looks good. So the block of glycolysis, it can be actually bypassed by adding acetate to the cells, right? Now let me talk a little bit about the other branch, right? Fatty acid synthesis. So we look at the enzymes involved with fatty acid synthesis also, and what we saw was a downregulation in both RNA level and protein level in both treatments with IN1 and KIR. So this pathway is also downregulated. It makes sense because acetyl-CoA, it's a key molecule common to both pathways. If we now bring down the pool of acetyl molecule, the amounts of acetyl molecule inside the cell, it makes sense to us to observe this downregulation. This downregulation, it's again, correlates with cell differentiation in the kidney. I want to call your attention here for the lyase in special, acetylyase, ACLY, which is a main enzyme that converts the acetate, the citrate into acetyl-CoA outside the mitochondria. So what we did, we shift gear to a more genetic in vivo approach, and we knock out the acetylyase in the nephron progenitor cells only. It's a tissue-specific knockout. And what we've seen here, let's look at the two panels on the left side, and what we see here, the mutants have, look at the white staining, the mutants have a smaller cap with fewer cells in the absence of, in the knockout cells, the ACLY knockout cells. And if you look at now on the right side, on the right side of the screen, the mutants, they have a lot higher expression of Wnt4. And Wnt4, it's one of the main markers for differentiation in the developing kidney. So, so the ACLY knockout clearly brings down the levels of acetate inside the cell, and this drives cells into differentiation. I'm showing you this also in vivo here. So the main points of these thoughts. First, glucose metabolism mediates cell fate decisions in the developed kidney, as I showed you throughout the whole presentation. Glycolysis inhibition, as well as KIR treatment, or activation of the canonical Wnt pathway, promotes downregulation of the cholesterol biosynthesis pathway, as I showed you. And both glycolysis inhibition and KIR treatment decrease acetyl-CoA synthesis in NPCs. Statin treatment, which is again, pravastatin, the drug that blocks cholesterol, the cholesterol pathway treatment, increased nephrogenesis, analogous to what we're seeing with glycolysis inhibition at the beginning of the presentation. So acetate appears as a key metabolic checkpoint that regulates cell fate decision in NPCs, nephroprogenitor cells. So there is a clear link between cell metabolism and cell fate decision in the developing kidneys. I'd like to take a few seconds to acknowledge people in my lab, our collaborators, and the founding agents without which we'll be able to do this work. I'll stop now and take questions if any. Thank you very much for your time and attention today.
Thank you, Giovanni, for the beautiful presentation. A lot of nice work here. We have a couple of questions, and one is, when you inhibit glycolysis to induce differentiation, is glutaminolysis and or beta oxidation increased?
Yes, we are conducting a number of experiments in vivo right now to sort out this issues. The thing is, the progenitor cells, they do not metabolize lipid very well, okay? Although, having said that, we're looking at long chain fatty acids. We have to look back to analyze medium and short chain fatty acids, but beta oxidation is increased in those NPCs when we block glycolysis, but we have to sort out better this data.
Okay, thank you. And then we have another question that says, if oxalate transport can play a role in NPC.
Oxalate transport can play a role in NPC. Every single metabolite in the cell could play a role for NPC metabolism. Mainly when we knock out, when we decrease the flux of the glycolytic pathway, then it could become more important. One thing that may, if I understand correct the question, so the pyruvate can also be converted into oxaloacetate using a carboxylase, pyruvate carboxylase enzyme, and that would increase the levels of oxaloacetate inside the mitochondria and helps out to move forward with the Krebs cycle. I don't know if I answered the question, but thank you.
Thank you. Then someone's asking, have you looked at mitochondrial oxidative phosphorylation in this study, and does that show any alterations to affect cell fate determination?
Yeah, we're looking at that right now. So we have collecting data, and we already have a bunch of data looking at the mitochondria specific metabolism, and it does play a role for cell fate decisions. Yes. Okay,
great. We have time for maybe one or two questions more. When you inhibit glycolysis, what happens to increased amount of glycolytic metabolites?
When I inhibit glycolysis, I decrease glycolytic metabolites, not increase.
Okay, thank you. Which of the, this will be the last one, I think. Which of the presented pathways contribute most to NPC cell fate?
Oh, so we see acetate as a key molecule that is in the cross point, in the crossroad, if you will, and that feeds into different pathways, and we analyze them. But clearly, the cholesterol pathway seems to be a big, play a big role for cell fate decision in those cells.
Okay, Thank you. I think that's all we have time for, but you have more questions that you maybe can answer later on. So
thank you very much. So I want to thank everybody for being part today here with us in this meeting. It's a new format. And thank you very much for your collaboration, time, and attention. And I'll thank you very much the organizers for putting together this meeting today. Thank you.